Alright beta, let's go ahead for a little bit more details further. Now what are the further details that we need to understand? ये सारे हमें केमिकल रिएक्शंस के डिटेल्स दिए गए हैं जो हम ऑलरेडी डिस्कस कर चुके हैं. तो ग्लाइकोलिसिस के प्रोडक्ट में क्या आ जाएगा, हमें मिल चुका है ATP, NADH2 एज वेल एज पायरुविक एसिड. ये जो पायरुविक एसिड मिला है, अनफॉर्चुनेटली द आउटर मेम्ब्रेन ऑफ माइटोकॉन्ड्रिया इज नॉट परमिएबल टू पायरुविक एसिड. सो सीन क्या होता है कि अगर मेरे पास ये माइटोकॉन्ड्रिया हो गया कुछ इस प्रकार से, तो जो पायरुविक एसिड है वो आउटर मेम्ब्रेन से तो पास हो जाता है, लेकिन इनर मेम्ब्रेन जो होता है उससे पास नहीं होगा, बिकॉज इनर मेम्ब्रेन, सॉरी आई सेड आउटर, जो इनर मेम्ब्रेन होता है दैट बिकम्स इम्परमिएबल फॉर माइटोकॉन्ड्रिया. सो इन बिटवीन ओवर हियर यू हैव pyruvic acid getting converted to acetyl molecule, that is acetyl coenzyme A. This reaction is called as link reaction or transition reaction. It can also be called as oxidative decarboxylation reaction. Ideally, originally, this occurs in intermembrane space or intermitochondrial space, or intermitochondrial membrane space. But NCERT कहता है कि ये माइटोकॉन्ड्रिया के मैट्रिक्स में हो रहा है, तो हम कहेंगे ठीक है माइटोकॉन्ड्रिया के मैट्रिक्स में हो रहा है. ऑलराइट, व्हाट इज द रिएक्शन दैट इज अकरिंग एग्जैक्टली? तो यू विल हैव pyruvic acid. Okay, I'll show it in the better way over here itself. You have pyruvic acid that tends to get converted into acetyl coenzyme A, and this conversion that we are observing, is possible because there is release of carbon dioxide as well as there is release of hydrogen. The hydrogen that is released is taken up by NAD and gets converted to NADH2. Along with that, there is release of carbon dioxide and there is addition of coenzyme A. गलत बोल दिया ना. There is release of carbon dioxide and there is addition of coenzyme A. There is also release of hydrogen ion which has been accepted by NAD to form NADH2. And this entire reaction is being catalyzed by an enzyme which is pyruvate dehydrogenase. पायरुविक एसिड से हो रहा है तो नाम आ जाएगा पायरुवेट डिहाइड्रोजनेस. ऑलराइट बेटा, आई होप द कॉन्सेप्ट इज क्लियर. सो व्हाट ऑल थिंग्स आर हैपनिंग ओवर हियर? Pyruvate dehydrogenase enzyme is responsible for converting pyruvic acid into acetyl coenzyme A, and during this reaction it removes hydrogen from the pyruvic acid as well as carbon dioxide from the pyruvic acid. Alright, and it adds coenzyme A. It adds hydrogen to NAD basically. Now this becomes your Krebs cycle or tricarboxylic acid. Right, let's try to understand their basics very firstly beta. तो ये जो TCA cycle है या फिर Krebs cycle जो, it was very firstly been discovered. It was discovered by Sir Hans Kreb. Right, Sir Hans Kreb was the scientist who basically discovered this particular cycle. This occurs in the matrix of mitochondria. माइटोकॉन्ड्रिया के मैट्रिक्स में ये साइकिल परफॉर्म होता है. इसके अलावा ये साइकिल जो है इट अकर्स ओनली इन एरोबिक रेस्पिरेशन. सिर्फ और सिर्फ एरोबिक रेस्पिरेशन में ये साइकिल होता है, बाकी किसी भी रेस्पिरेशन में ये साइकिल नहीं होगा. दिस साइकिल can have the capability of providing 30 ATPs, and In them, the first stable compound that is formed is nothing but citric acid. इसका अगर केमिकल फॉर्मूला देखेंगे हम तो इट इज दिस. There are 3 carboxylic acid which are present in it, and because of this specific thing you also call the cycle to be as tricarboxylic acid cycle. Alright beta, or you can also call it to be as citric acid cycle. Alright, I hope it is clear up till here. Let's try to understand this entire cycle that is given over here. यहाँ पे काफ़ी सारे steps बीच में skip किए हैं, हम वो सारे steps पढ़ लेंगे बेटा. Let us understand all those steps. We will try to understand all those steps. So let's just try and understand them. Alright. Now, when we talk about this, very firstly we will have link reaction that will be seen. That is nothing but pyruvic acid getting converted to acetyl coenzyme A. ठीक है, pyruvic acid will get converted to acetyl coenzyme A, wherein there is conversion of NAD into NADH2, as well as there is removal of carbon dioxide and addition of coenzyme A. The enzyme involved to do this reaction, as we know, is nothing but pyruvate dehydrogenase. It is nothing but pyruvate dehydrogenase. इस एसिटाइल कोएंजाइम ए के साथ कंबाइन हो जाता है oxaloacetic acid. You have combination of oxaloacetic acid which will combine with acetyl coenzyme A का सिर्फ acetyl group and get converted into citric acid. It will get converted to citric acid. Alright. This happens with the help of an enzyme called as citrate synthase, wherein there will be removal of coenzyme A, that is nothing but added back to the reaction. You will have addition of water also over here. This citric acid further gets converted to its isomeric form to form isocitric acid. However, when you have formation of isocitric acid, in this case you have cis aconitate नाम का एक अनस्टेबल कंपाउंड. ये जो 6 अकोनेटेट नाम का एक अनस्टेबल कंपाउंड है, ये पूरे रिएक्शन को कैटलाइज करने के लिए एक एंजाइम होता है जिसका नाम है अकोनेटेस, जो सबसे पहले सिट्रिक एसिड में से पानी निकाल देता है, तो बन के मिलता है सिस अकोनेटेट. फिर वापस से रीअरेंज करता है इस सिस अकोनेटेट को बाय एडिशन ऑफ वाटर to form something called as isocitrate. Alright, this isocitrate which is formed, it gets converted to oxalosuccinic acid. It gets converted to oxalosuccinic acid. This happens by the removal of hydrogen molecule, and the hydrogen molecule that is removed is been accepted by NAD to form NADH2. अब जब हाइड्रोजन रिलीज हुआ है तो ऑटोमेटिकली एंजाइम का नाम हो जाएगा क्या बेटा? करेक्ट, आइसोसीट्रेट, क्योंकि उसमें से निकला है डिहाइड्रोजिनेस. You've got isocitrate dehydrogenase as the name of enzyme. This oxalosuccinic acid what we have, further gets converted to alpha keto glutaric acid. It gets converted to form something called as alpha ketoglutaric acid, wherein there will be removal of carbon dioxide, and the enzyme involved is oxalosuccinate decarboxylase. Correct. The enzyme is oxalosuccinate decarboxylase. This alpha ketoglutaric acid further gets converted to succinyl coenzyme A. Now, while it is getting converted to succinyl coenzyme A, the reaction is pretty much similar to what we've understood earlier. यहाँ पे जो हमने किया वही सेम चीज़ यहाँ पे हमें करना है, which is what there has to be removal of hydrogen, which is taken up by NAD to form NADH2. There's got to be release of carbon dioxide as well. There is CO2 which is released during this case. थोड़ा messy हो गया ना, clean कर देते हैं. There is removal of CO2 as well as there is addition of coenzyme A. Alright. The enzyme involved over here will again be same, क्योंकि hydrogen release हुआ है तो enzyme's name will be correct. Alpha ketoglutarate dehydrogenase. Enzyme will be alpha ketoglutarate dehydrogenase. Succinyl CoA, which is so formed, will now get converted to form succinic acid. अब इस कन्वर्जन के दौरान एक काफ़ी यूनिक चीज़ होता है, वेरिन एनर्जी तो रिलीज़ हुआ, बट ये एनर्जी को इस्तेमाल किया जाता है फॉर फॉर्मेशन ऑफ GDP, यानी कि GDP guanosine diphosphate will take up this energy to add one more phosphate group and form GTP. However, majority of the time GTP is not utilized as energy currency by the cells, and that is the reason this GTP immediately again gets broken down, and when it gets broken down, the energy that is released is taken up by ADP to form something called as ATP. Alright, and the enzyme involved in doing this entire reaction is called as succinyl thiokinase. बेटा इस पर्टिकुलर स्टेप में जो ATP बना है वो एक सबस्ट्रेट को दूसरे सबस्ट्रेट में कन्वर्ट करने की वजह से बना है. तो जब इस प्रकार से कोई एनर्जी बनता है जहाँ पे एक सबस्ट्रेट को दूसरे सबस्ट्रेट में कन्वर्ट किया जाता है, तो इस प्रकार हम कहते हैं सबस्ट्रेट लेवल phosphorylation. So this reaction is identified to be as substrate level phosphorylation. Substrate level is because one substrate is getting converted to another substrate, and phosphorylation is because ATP is synthesized during this stage. Alright. Succinic acid which is so synthesized right now, अभी जो succinic acid synthesize हुआ है, it gets converted to fumaric acid, और इस कन्वर्जन के दौरान again hydrogen released is not taken by NAD but instead is taken by FAD to form FADH2, and the enzyme will again be dehydrogenase. If you remember, during biomolecule we've discussed this concept that you have malonate as one of the compound which is similar to succinic acid that binds to this dehydrogenase and thus does not help in conversion of fumaric acid, and malonate acts as competitive inhibitor. So it is this reaction only that it inhibits. Alright. fumaric acid which is so formed gets converted to malic acid. During this conversion there is addition of water molecule. Here I've missed out, you have removal of coenzyme A which again gets done over here. Enzyme for this step, इस वाले step में जो enzyme involved रहेगा that will be fumarase. And lastly malic acid again gets converted to oxaloacetic acid, wherein there is removal of hydrogen taken up by NAD to form NADH2. The enzyme is dehydrogenase. The enzyme is dehydrogenase. So this is nothing but entire Krebs cycle. Yeah, there you go. This becomes your entire Krebs cycle that you will need to understand for your NEET examination. अब अगर यही चीज़ हम यहाँ पे देखेंगे बेटा तो काफ़ी सारे इंफॉर्मेशन मिसिंग है इसमें, because of which it will become difficult for us to understand or give the statement based answers in them, and thus we've understood this in very detailed manner in this. So कोशिश करो कि आप यही रेफ़र करो अपने नीट एग्ज़ाम के लिए, ठीक है. I'll just give you some space, you can take a screenshot of this, ठीक है. Remove me, if you want you can take a screenshot. Alright, let's go into detail further. अब आगे ये क्या कह रहा है हमको, ये कह रहा है कि सारे एंजाइम्स जो क्रेब्स साइकिल के लिए हमें चाहिए वो माइटोकॉन्ड्रिया के मैट्रिक्स में ही मिल जाते हैं, एक्सेप्ट सक्सिनेट डिहाइड्रोजनेस जो उसके इनर मेम्ब्रेन में होता है, ना कि मैट्रिक्स में, ठीक है. इसके अलावा ऑक्सालोएसिटिक एसिड इज़ द फर्स्ट मेंबर, नॉट मेम्ब्रेन, गलत लिखा है, इट इज़ द फर्स्ट मेंबर ऑफ क्रेब्स साइकिल, ठीक है. एंड क्रेब्स साइकिल has different things that we need to understand. There is one substrate level phosphorylation which is ATP synthesized. You have 3 NAD and 1 FADH2 which has been synthesized, wherein this becomes its net reaction. This is the net reaction where pyruvic acid along with 4 NAD, 1 FAD, 2 water molecules and ADP forms 3 carbon dioxide, 4 NADH, that is 4 NADH2, 1 FADH2 and 1 ADP which has been formed. Alright, how many ATPs obtained in Krebs cycle? 30. Half 30 sir, let's try to calculate. So energy calculation करेंगे तो हमारे पास एक ATP तो बन ही रहा है, वो तो sorted है बेटा. Total number of ATPs formed is 1 during this reaction. NADH2 जो है, एक NADH2 3 ATP दे रहा है हमको, और ऐसे हमारे पास 4 है. So you have 12 ATPs formed over here, 4, ठीक है. You've got FADH2, एक FADH2 हमको 2 ATP देता है और ऐसा एक ही है हमारे पास. You have 2 ATPs. So 12 plus 1 that is 13 plus 2 that is 15 ATP. So one Krebs cycle if when happens, you've got 15 ATP which is formed, out of which 1 is formed corely in it, remaining 2 are formed in ETS, ठीक है. However, aerobic respiration के दौरान glycolysis 2 बारी होता है because glucose is the substrate. So then you multiply this with 2 and you get 30 ATP as your answer. Alright, and that's how Krebs cycle से हमको 30 ATPs मिलता है. तो 8 हमको glycolysis से मिला, 30 हमको यहाँ से मिल गया. I hope the concept is clear बेटा. So that's nothing but your Krebs cycle. Let's go ahead and try to understand a little bit more details. When you talk about aerobic respiration, glucose, fructose will provide us with 38 ATPs. Sucrose is combination of these two and thus you get 76 ATPs. If we start from phosphoglyceraldehyde, we get 20 ATPs. If we start from phosphoenol pyruvic acid, we will get 16 ATPs. If we start directly from citric acid, we will get 12 ATPs. And if we start from succinic acid, which is way later, we will get only 5 ATPs. Alright, I hope the concept is clear so far.